You can start.
Good afternoon to all the participants present in this conference. Let me take this opportunity to introduce myself as Premraj Kashyap. I am the co-chairman of Quality and Standard Subcommittee of West Indies and CII. At the outset, I would like to extend my warm welcome to our esteemed speaker, Dr. Purushottam Sutar, Product and Business Development Manager, Hitachi High-Tech India Private Limited, and Dr. Nivedita Patil, Assistant Manager Application Specialist, Hitachi High-Tech India Private Limited, who have kindly joined us today to share their expertise and insight on this important and evolving subject. I would also like to acknowledge and thank Hitachi High-Tech India Private Limited, our knowledge partners, for supporting this program and also for bringing valuable technical expertise to this discussion. As we commence the activities of CII Western Region Subcommittee on Quality and Standards, I would like to briefly share a broader vision that guides our work. For us, the starting point is very clear. Quality can no longer be viewed merely as a matter of compliance. In today's competitive environment, quality is increasingly a driver of productivity, operational excellence, customer trust, innovation, and global competitiveness. As Indian industry become more integrated with global value chains, our ability to consistently deliver high quality products, processes, and service will be critical to our competitiveness. Therefore, our endeavor through the committee is to help industry move towards a mindset where quality is not seen simply as a requirement, but as a strategic business enabler. A major focus of our efforts this year is strengthening quality capability among MSME. MSMEs are the integral part of India's industrial ecosystem, but many continue to face challenges to understanding standards, building quality systems, and navigating compliance requirements. Equally important will be our efforts to strengthen the industry regulator interface, particularly around quality control orders, QCO, and regulatory standards. We recognize that the effective implementation require both greater clarity from regulatory institutions and constructive feedback from industry as well. The committee is therefore working towards creating a meaningful platform for engagement with BIS, QCI, and other quality institutions while facilitating structured industry feedback on implementation challenges. Along with this, we are also focusing or encouraging wider adoption of globally recognized quality management practices such as lean manufacturing, TPM, Six Sigma, Kaizen, and other quality excellence methodologies. We also believe that one of the most effective ways of accelerating quality excellence is through learning from successful implementation within industry itself. For us, success will not simply be about the number of programs we conduct, but the real measure of success will be the impact we create. With this, I look forward to the active participation, ideas, and collective contribution of all stakeholders as we work together to make quality a stronger driver of business excellence and industrial competitiveness across the Western region. Thank you.
So I'll request to invite the speakers.
With this, I would request our esteemed speaker of today's conference, Dr. Purushottam Sutar, Product and Business Development Manager, Hitachi High-Tech India Private Limited, and Dr. Nivedita Patil, Assistant Manager Application, Hitachi High-Tech India Private Limited.
Yeah. Good afternoon. Good afternoon.
Good afternoon.
Yeah. So shall I take over?
Yes, sir.
Please.
Yeah. Okay. Yeah. Thank you, Rishna. Thank you, Premraj sir. So I'll just go directly to my presentation. So we will our team has divided the presentation in two parts. So I'll talk about the product, and later my colleague, who is the application in charge with Hitachi High-Tech, will take over and talk about more on the depths of the applications relevant to the peptides. Okay, so with this, I'll just switch off my video and I'll go to the presentation mode. Please let me know if my slides and everything is visible so that I can quickly start. Okay, and sharing the window, right? This one. Okay, so please help me if my screen is visible.
Yes, it's visible.
Yeah, okay, yeah, thank you. Yeah. So, so this is the topic for today that we will conduct is GLP-1 peptides in the spotlight. So the idea is decoding the therapeutic peptides and pharmaceutical applications with a very specialized instrument called amino SIA LA88080, which is again the amino acid analyzer from Hitachi High-Tech. So we have two presenters today. Myself, Dr. Purushottam Sutar. I am the senior manager for sales and support in India. I look after the spectroscopy and the chromatographic solutions, that part of the business in India. And with me is Dr. Nivedita Patil, who is the assistant manager and application specialist for the same division in my team. Okay. Going to Before going to the actual slides, let me please introduce our company. So this is Hitachi High-Tech. We belong to Hitachi High-Tech Group. It is named. It is basically divided into four groups: value chain, nanotechnology, analytical and medical solutions, and core technology solutions. So Hitachi High-Tech has presence in 26 countries with over 46 offices. The corporate office is located in Tokyo, while the factories are at Oyama Works and Naka Works. Hitachi High-Tech Science Corporation. So we are the manufacturer of all the analysis measurement equipment of Hitachi High-Tech Group for and in charge for development, production, sales, and services of all these products. In India, the head office is in Delhi, with sub offices in Mumbai and Bangalore, and we also have our application. The Center of Excellence in Bangalore and in Mumbai. So we have two demo facilities and multiple offices in India. So we are spread across the country. This is the product lineup that we have. Uh, so if you can see, we cater to all types of product from surface analysis, including the microscopy products, then elemental and physical property analysis. Then we have the spectroscopy and separation analysis. So the topic of interest today falls into this separation analysis category, specifically the amino acid analyzer. However, we deal in the entire range of the product. So coming to this particular model, we have nearly 60 years history in amino acid analysis production. The first model, that is the KLA2, was introduced in 1962, and it could do the one sample in 22 hours. However, with over the period of many years, we have developed the expertise and made some innovation and advancement in the technology to come up with the latest model, which is the LA8080 amino SIA. So the feature is that we can do the same analysis in 30 minutes, courtesy to the particle size reduction from 40 micron to 3 micron, and that's how we could give a high throughput and high productive instrument. Okay, coming to the instrument for today's discussion, this is the amino acid analyzer LA8080 amino SIA. It basically adopts a post-column ninhydrin method with a simple pre-treatment and high stability. So post-column ninhydrin technique is one of the gold standard technique in amino acid analysis. The instrument has a very good reproducibility, high data reliability, and precise quantification, and we can cater up to 40 or more amino acid analysis in this particular instrument. So we do have ready to use methods available for that. And one of the key features which is more relevant to GLP analysis is that this instrument also caters or has the software capability to comply with the FDA, US FDA 21 CFR Part 11 requirement. Okay. And coming to key specification, so it has a detected limit of 2.5 picomole, which is the lowest. A uh, direct injection mode facility, which helps to reduce your carryover. It's a better injection way of doing a sample injection. Sample capacity can be 100 or it can also scale up to 120. Reproducibility is very good in terms of both retention, peak retention time, and peak retention area. Coming to the actual face of the system, so this is how the system looks externally and internally. It is more or like similar to the regular HPLC system. It has the reagent box, what we also call as the eluent chamber. Then you have the auto sampler, then you have the pump. Pump is divided into two parts, pump one and pump two. Then you have the reagent box here, which has the ninhydrin and its stabilizing buffer. Then again, if you go to the top of that, there is a column oven, which is again a heart of any chromatography system. Similarly, here you have the column for separation. After that, you have the reactor, which is again one of the critical part of this system, which enables the detection of the amino acids. And then you have the detector, which is basically a UV detector, and then a nitrogen gas panel. So the entire system is kept into nitrogen. So you need some regulators to control it. So this is how the system overall looks like. Coming to each features, so the reagent box basically accommodates the buffers or the eluents, uh, which are then taken up by the pump. So pump one is basically for the eluent buffers, and after degassing, the eluent is then sent into the auto sampler and column, and subsequently to the system. So we can cater to a flow rate setting of up to from 0.001 to 1 ml per minute. It can withstand a pressure of 34 megapascal, and we can have a gradient elution up to we have 7 ports, so you can use 5 ports for the gradient elution, and one is for the reagent or the one is for cleanup, and another is for water. Again, which water can we use for cleaning as well as adjusting the gradient. So we have 7 mobile phase ports on this system. An auto sampler, we can give up to two variation. One is non-coolant and coolant. Both are available, but generally considering the peptide analysis, we prefer the coolant model with a 100 vial capacity, and you can accommodate your regular 1.5 ml vial in this. So no specialized vial is required, and you can go up to zero point 0.5 microliter to 100 microliter injection volume. The column heating is a Peltier based system. We can go up to 90-degree column on setting with a step of 1 degree, and the column which is used is a Hitachi custom ion exchange resin, only available with Hitachi. So that is something we have developed to have the best performance from the system. Then you have the reactor. So reactor is basically, it is based on electronic heating with a temperature setting of up to from 50 to 140 degree. This is where the actual reaction between the ninhydrin and the analytes happen. I'll explain it in the next slide. Then we have the detector. Detector is basically a spectrophotometric detector, and it is a aberration corrected concave diffraction grating detector, stationed at two wavelength, 570 nanometer and 440 nanometer. Okay, and then you have the reagent ninhydrin reagent box, which has the provision to keep the buffer as well as stabilizing buffer as well as the ninhydrin reagent. Okay, so this is an overall picture of the system. Coming to the working principle here, so as explained earlier, it is basically an amino acid analyzer which based on the cation exchange ninhydrin post column method. So if you see the pump one delivers the buffer, in this case citrate buffer. These are the eluent buffers into the auto sampler, into the column one, and to the reaction unit and the detector. So this is the path of your mobile phase or the eluent, in which you inject your sample. in via the auto sampler. So no manual injection, it is the auto sampler, some predefined microliter, after which the separation of the non-derivatized amino acid takes place. So until here, there is no derivatization. It is typically based on the non-derivatized amino acids. So since amino acids are non-chromophoric, to enable the detection in this stage, your ninhydrin reagent is pumped in via the second pump, and the reaction happens instantaneously at around 135-degree temperature, and then it imparts the chromophoric property to your analytes, which is the amino acid, and subsequently they are detected in the detector at 570 and 440 nanometer. Okay, so this is overall how the instrument operates. Yeah. And some of the key features of amino saya or this instrument is our reagents are commercially available. So there is no proprietary policy from Hitachi that you have to buy reagent from us. It's you can buy it from the open market. We do have certain recommendations where you can buy the reagents from the vendors. Additionally, Hitachi also provides the recipe of the buffers and reagent preparation. So the after sales or usage cost considerably reduces. So this is one of the key advantage of owning a Hitachi amino acid analyzer. Then automatic warm up. So it is equipped with an automatic warm up program as a standard. So in the standby mode is in this particular method, it ensures that the system is conditioned and equilibrated prior to initiating any sample analysis. This includes the pre-check of the analysis condition, including the flow rate, oven temperature, reaction unit temperature. So all variables and parameters are checked in the automatic warm-up, and system is brought to a start condition where you can directly do the analysis. Then there is automatic cleaning feature. So this is again a standard feature where if there is some interruption in your analysis instrument operation, then automatically the cleanup method is triggered, and whatever is remaining in the system line, it automatically cleaned up. This is again a feature which user doesn't have to do anything, it is automatically triggered. And then prevention of the reagent deterioration. So usually in amino acid analyzer analysis, you need to mix the ninhydrin reagent with the buffer, and then you take it out and preserve in the fridge. So all those manual activity is not required. You simply install the ninhydrin reagent and the buffer. You can see the two bottles onto the system and keep it there as long as you want to use it, or they get exhausted. There is no deterioration of the reagents thanks to the system which is developed in such a way that the entire system is kept under nitrogen gas to ensure that it has the reagents has a good life, long life. Okay. Coming to the column, so Hitachi is the first manufacturer to introduce and use 3 micron particle size column. This reduced particle size has enabled to achieve ultra fast analysis within 30 minutes, and which is the fastest among the competitor, ensuring the high resolution of 1.2 within 30 minutes. This can be seen from the chromatogram. This is our standard chromatogram, which is showing the separation of the naturally occurring 18 amino acids. Okay, this is for a reference. And going further, the concept of our instrument is basically defined on these parameters to ensure comfortable operating conditions. So the ergonomic desktop model was designed in considering the operability and flow lines are at operator's eye level. So user can comfortably open the system and handle. No need to climb any stairs or something. So it is basically very easy for the user to use it. And many users nowadays are very used to the HPLC kind of setup. So practically this system is based on similar concept. Okay, then we have the software. This is a user-friendly screen, user-friendly status screen. So operators can easily find and check the status of each part, various parameters, and the manual operation and the maintenance interface are also placed here. So everything is sitting in one screen. So you don't have to toggle between multiple screen for usage. Then this is the place where uh, since amino acid analysis takes a longer time, the optimum usage of your reagent is very important. So this is a feature where the automatic reagent amount calculation helps to basically understand how much reagent is required. So during your analysis, there should not be a situation that the reagent is exhausted or you overuse anything. Okay, so it ensures the optimum usage of your system and good performance maintenance of the system. Okay. Then compact. So this is something from our previous generation. The instrument is designed in such a way that we have reduced the area which is occupied by the instrument by 30%. So the lab space is optimally used. Then use of high reliable and stable post column methods. So over the years, we have introduced many models for amino acid analysis, and over these past years we have developed a lot of intelligence and methods for the various models, and all those has been incorporated into the new models. And in fact, the methods and columns which were being used for earlier model can straight away use as such on the new system as well, and can be reproduced easily. Okay, coming to this next feature is this is the reaction module is an important part of this instrument. Any make, any model. This is where the reaction happens and your sensitivities and reproducibilities of your analysis is defined. So competitors, they adapt a reaction coil, whereas we have developed our own reactor called the thermodynamic efficiency, the third reactor. So by virtue of this, our system can provide highest sensitivity analysis with the best efficiency for this particular method. So features is since we use this particular technique, we could achieve a detection limit of 2.5 picomole. Previously, this was 3, so we have still gone down. Improved thermal conductivity, which can also help to analyze di and tri peptides with high level of sensitivity, and the long life. This particular reactor can have an average life of up to 5 years. Previously, I mean, it was 2 years, but we can go up to 5 years as long as the flow path is not clogged, which again the system also takes care by automatic cleaning process. But some level of maintenance, if you use the system properly, the life of the reactor is very long. And this is something in computer you need to keep on really changing, and it involves some cost. Okay, so considering all these features, so this is the figure that I want to emphasize, that it shows the enhanced sensitivity due to the usage of the thermodynamic TDE3 unit as compared to the conventional reaction column. So overall, we have observed that there is a 5% increase in sensitivity for the pH method, which is the conventional 18 amino acids method, and 26% increase in the free amino acid analysis, which is the physiological fluid methods analysis on our system. Okay. Coming to an important part, which is the compliance. So the system operates on the Open Lab software, which is a very well known 21 CFR compliant software. So this is how the screen looks. So you have a screen for the data acquisition, then you can have the reports, then diagnostic, audit trail, everything is available as such. So an HPLC user basically knows what software is, and similar features are available here. What we offer is ready to use method. So there are two methods which we offer, which are ready to use along with the column. The protein hydrolyzation analysis method. This is basically for the naturally occurring amino acids, which is 20 in number, mostly catered for the nutrients and the fertilizer feedstuff analysis. And then we have the physiological fluid analysis method, which is basically for the free amino acid. We can go 40 or even more, and aimed at the food and biological sample. So these are the two methods that we offer here. And to cater to these two methods, we have various columns which are available. Again, the choice of column depend on the runtime or the separation that the user wants. So we have multiple column available, but yes, we start with the standard columns with 30 minutes of runtime, and then as per need, we can provide the various columns as well. Okay, so to summarize this particular slides, these are the advantages that we see with our Hitachi amino acid analyzers. The operations are automatic. You just need to prepare the sample and put it on the system. Derivitization, separation, everything is done by the system. It depends on a highly stable derivatized analytes, ninhydrin method, which is again developed in 1958. It is still the gold standard method, better than the pre-column method. The column has a very long life. It has a highly efficient TD3 reactor, which nearly gives you a life of 5 years. A very simple sample prep, which is used here, can cater up to 50 amino acids. Cost per sample is low. High reproducibility, stability, resolution, and separation efficiency we can offer, along with the compliance to the standard testing methods and regulation. So this was a concept about the product. Now we would like to move on to the applications part of this, and I would like to hand over my mic to my colleague, Dr. Nivedita, who has done a lot of work on the peptides, and she can really throw light on what application and what are our strength in this area. So yeah, Nivedita.
Good afternoon, everyone. Thank you, Purusan, for the wonderful product presentation and explaining the features, technical features of AminoScience. Now we will move on to the capabilities of AminoScience. So to begin with, I will start with the basic chemistry or the just a refreshing our what is amino acids. So amino acids are generally considered as a fundamental building blocks in the origin of life theory, abiogenesis, and as they form protein. The DNA and mRNA, DNA and RNA code the proteins, and yes, which are again made up of amino acids. So they are the molecular machines of the living cell. And to analyze these molecular machines, amino acid analysis is the key technique, or we are mainly a gold standard technique to analyze these amino acids. So as I said, the chemistry. So what is an amino acid? As the name itself suggests, amino group and the acid group. So, and the molecule which has this amino and the acid group, but the R part makes it different. Like if you can see in glycine, it has H. R group, again, the imino group, the taurine. So differing in the R group, the chemical structure changes, the name changes. Now, moving forward, as I said, amino acid is a molecule which has amino as well as acid group. Depending on the substitution, the R group which is present, we can divide them into aliphatic amino acids, hydroxy or sulfur containing amino acids. There are some less cyclic amino acids, aromatic amino acids, basic amino acids, and acidic and their amide groups. So our body composed of 20 amino acids, and apart from that, there are another amino acids which we call as a non-proteogenic amino acids, which are 40 or more, which are found in our blood. So to understand again much more better the technique and its application, first understand this ion exchange chromatography was invented by Stein and Murray, for which they were awarded a Nobel Prize also. And this has, from there, it has become an international standard method for amino acid analysis for qualitative as well as the quantitative analysis. So as Purusan has explained that it is based on the cation exchange chromatography with post-column derivatization technique, just understanding of, as you can see in the figure, based by changing the pH of the eluent, the salt concentration, and the affinity between the amino acids and the resin or the column pack material can be changed and the separation can be achieved. As amino acids lack chromophore, hence, just monitoring the pH and the ionic strength, the separation can be achieved on the our Hitachi patented column. So, as you can see, our chromatogram having a different, based on their different acidic groups like acidic amino acids, neutral amino acids, and the basic amino acids. So you can see the profile of the chromatogram in the way in which they are eluted. So just a brief that yes, Purusan has very well explained the principle of amino saya. The buffers and the reagents help in the separation of this. The buffers help in the separation of amino acids as there are no chromophore attached. The separation of the amino acids is achieved, and the reagent helps to enhance it to a wavelength of the visible detector at 570 and 440 nanometers. Now, moving to why amino acid analysis, or why this technique has become the gold standard for peptide and protein characterization. As I have explained that every peptide and protein is a fundamental sequence of amino acids, and amino acid analysis or amino acid analyzer will give the idea about the composition of each amino acid, help in determining the content, what is present, verify the recovery, confirm the expected count which is required, and also help in the sequence verification through the residual count which we obtain. So we do this hydrolysis under acidic medium for most of the amino acid as the procedure described under USP, then separation and quantification. So this is one of the standard chromatogram, as you can see here, amino profile, and then you get the content, the sequence verification, recovery, calculation, and the composition of the respective analyte. So it matters because all of these features can be achieved with the amino saya under regulatory compliance. Moving forward now, why amino acid? And yes, amino acid analyzer has been or analysis method, that is the ion exchange chromatography with post-column derivatization, is a gold standard. But why peptide therapeutics? Okay, so in olden days, mostly, or in the ancient days, small molecules were preferred to that of the peptide therapeutics because of the stability of the small molecules. But now, with the evolution or revolution, since the peptide therapeutics has revolutionized modern medicine through structural engineering, their stability, efficacy, and half-life has increased. And the examples of these is the blockbuster drugs, semaglutide and tirzepatide. With their billion-dollar sales, we can see that over the period of time, the pipeline of the peptide therapeutics has increased, and so the stability and efficacy. But this has increased also and challenge analytical challenges to study these molecules, and that's why amino acid analyzer is needed to trace this modern peptide therapeutics. So from the application point of view, in today's webinar, we will be exploring how amino saya will help in the USP compliant peptide therapeutic characterization, as well as the quality controlled GLP-1 peptide drugs, how using our different pHs, as Purusan has explained, we provide the recipes for the buffer, and we have 7 ports, and using that, how it can help in the method development and the research purposes. So we have shown one of the example of it. And again, the application towards the biotherapeutics, like how it monitors in the biomedicine production, as well as bioprotein therapeutics, amino acid analysis of protein therapeutics. So let's begin. So these are the USP monographs available for the peptides, leuprolide acetate, calcitone in salmon, and exenatide. So I have given the amino acid criteria requirement for that, which we will be again seeing in detail. So these are the 3 monographs for the peptides available, and we will see the capability of amino saya with these examples. So leuprolide acetate is a synthetic nonapeptide analog of natural hormone GnRH, that is gonadotropin releasing hormone, and it comprises of 9 amino acids with the modification at C6 position, that is D-leucine and N-ethylamine prolylamide at the C terminal. So if you look at the structure, it is just a 9 amino acid peptide, a simple molecule, but it presents an analytical challenge that required informed interpretation of data. So if you look at the glutamic acid structure, it is a pyroglutamic acid. So with the help of our sample pretreatment method, it can be analyzed or converted into the glutamic acid and you can analyze it. Similarly, the tryptophan, which is an acid sensitive. So most of the time when we treat the peptides, it degrades during acid hydrolysis. So with our pre-treatment method, we take care to show the recovery of the tryptophan as a present, and with a good recovery around 0.5 to zero point 0.9 count. Then, as I told, there is a terminal ethylamide modification which is happening at the proline. So again, the recovery of the proline is again, since there is a structural modification at the proline peptide, that can also be achieved. So these are the structural challenges which are present in leuprolide acetate, and that structural challenges has been overcome by aminosaya. With the help of aminosaya, as you can see, the counts, the calculated counts are within the accepted criteria, satisfying the USP compliance, which is given for the leuprolide acetate in the monograph. And we have also used the USP compliant analysis methods like sample pretreatment method with some modifications, which help us to achieve this or place the leuprolide within the accepted criteria. So moving forward, now our next example is, initially it was the 9 amino acids with a lot of terminal modifications in it. Now we will move towards the 32 amino acid peptide, again hormone, and again there is a monograph for this in the USP. So we have done the analysis as per the USP monograph, as since here the structural demands or the structure has the N-terminal disulfide bridge. Okay, so we need to analyze this molecule by another performic acid hydrolysis method, which is again mentioned in the USP. And with this, with the methods described in the USP, again our accepted observed criteria fall under the criteria which is mentioned in the USP. With the net peptide content 100%, molecular weight determined AA derived, we can say 99.94%, and the amino acid profile compliance with the expected stoichiometry. So again, aminosaya has shown its capability or demonstrated the capability for this another USP monograph peptide drug calcitonin. Now our next case study is exenatide. Again, now from 9 to 32, now we'll moving to 39 amino acids. It is a GLP-1 receptor agonist with again official monograph in the USP, and it is mostly an anti-diabetic drug. So here we have just carried out again acid hydrolysis and we have compared this amino acid composition or the observed count with that of the accepted criteria, and the observed ratio or the count show excellent agreement with that of the expected, confirming the peptide identity, highlighting the aminosaya's robustness to support the USP compliant identity testing and accurate quantification for amino acids and USP compliant methods. So these are some of the representative examples to showcase that whatever method which we are offering and our instrument and the features which Purusan has explained, we are compliant with the USP criteria. So what is the USP requirement if we look into? USP requirement is that the detection limit for each amino acid should be not more than 10 picomole. Again, for proline, it should be 50 picomole. The linearity should be determined between 200 to 500 picomole. Precision and repeatability should be not more less than 2%. Again, the resolution between the critical isomers pair, that is isoleucine lesion, should be more than 1.5%. And with this, we can say that the aminosaya falls or explains its performance with the limitations or the expectations exceeding the USP requirement. So we have successfully evaluated 3 USP monograph as per study as per 3 USP monograph for peptides exenatide, calcitonin, and leuprolide acetate. Now, as we have seen, there were not more structural modifications, and they were like a simple 20 amino acids, peptide amino acids which are there in a peptide. But nowadays, as I explained, the peptide therapeutics has gained a lot of importance for the drugs, like you can say semaglutide, terzeptide, as there is a lot of structural engineering has happened within the molecule. If we go from the oxytocin and insulin to the semaglutide and terzeptide, so to again to analyze this, or the analyst falls under a lot of pressure to detect the amino acids, as there is a lot of structural modification within these peptides. And yes, with this, we want to focus that even if the what your peptide is structured, or there is a modification in the peptides apart from these proteins, like proteogenic amino acids, even if there are non-proteogenic amino acids or fatty acids side chain, amino can deliver its best performance. So as I said, the efficacy and the stability of the peptides nowadays has increased by incorporation of the non-proteogenic amino acids, and there are more than 900 non-proteogenic amino acids still they detected, and AminoScience can help you to achieve the detection of this. So moving forward, as I said that the engineering with non-proteogenic amino acids is the key current trend of the peptides, along with the stability linker. So if you can see here in the structure, nowadays the peptide trend is following the linker and the stability enhancer, as the peptides are known to be overcome degradation. They're known for the degradation, or the half-life is very less. So that has been achieved by incorporation of the non-proteogenic amino acids along with the stability linkers. So incorporation of the non-proteogenic amino acid definitely has enhanced their half-life, as well as the efficacy and receptor selectivity. So this is the current trend of the peptides, and how AminoScience is going to demonstrate that analytical efficiency. Let's begin. So moving towards our GLP-1, again, it is a 31 amino acid peptide with, as I have shown, a linker and fatty acid side chain. So when we follow an hydrolysis procedure, with our sample pretreatment method, you can see the detection of all the amino acids which are present in the peptide, and there is no matrix interference from the side chain, that is a palmitoyl lipid side chain, and the detection of the tryptophan also. So liraglutide was accurately quantified using AminoScience, demonstrating no matrix interference from the lipid side chain. So this was again explained with the results. As you can see, the observed count was as per the expected count. And to another point, to bring to a focus, that there is a lipid linker in the structure. Okay, so that was also we were also able to trace that with our sample pretreatment and the analysis methods. So yes, again the 32 amino acid residue, 32 amino acid peptides was detected and matched the expected sequence of liraglutide. Then molecular weight was as per the AA, which is derived, it is around 374 daltons. The assay content was 102%, and the recovery which we obtained was 86.3%. Again, the EP compliant, or the USP, asked for the resolution for critical amino acid pair, that is isoleucine and leucine, that was around 1.69. So exceeding the this thing. Now let's move to another GLP, that is a second generation or current generation drug, that is a semaglutide. As I have explained, there are again current peptide trends have incorporation of non-proteogenic amino acids. So in this, there is an AIB and linker aminoethoxy acetic acid. So again, we have to see the matrix interference, the detection of from the lipid side chain, the detection of AIB, and the recovery of the peptide where, amino acid where this uh, linker or the lipid side chain is attached. So you can see we were successfully able to verify the sequence of semaglutide meeting the resolution requirement as mentioned in the pharmacopoeia with good recovery for tryptophan during acid hydrolysis and the detection of non-proteinogenic amino acid AIB and the linker amino ethoxy acetic acid. So that was also detected and quantified. So the observed amino acid sequence matched the expected profile with overall peptide recovery of 88.5%. And the analysis confirmed the detection and the quantification of non-proteinogenic amino acids. Also, so these results clearly demonstrate that the capability of AminoScience to analyze the GLP analogs with high reliability, compliance, and good recovery. So again, you can see the observed count is matching that of the expected one. And all the molecular weight, the sequence, the assay is in compliance with that of the in compliance with that of the or the expected one. So now, as I said that there is an incorporation of non-proteinogenic acid, and we have seen that there is no matrix interference occurred because of the fatty acid side chain when we analyze the peptides. The peptides are only, or the structural engineering is not only limited to one or two incorporation of non-proteinogenic amino acid like AIB. There are more non-proteinogenic amino acids within the peptide sequence for their stability purposes. So to demonstrate that, what we have done is we have prepared a custom standard with the incorporation of non-proteinogenic amino acids like AIB, alloisoleucine, nor-isoleucine, beta-alanine, ornithine. So we have incorporated 5 amino acids in our standard 18 amino acids. So this is the chromatogram of total 23 amino acids. As you can see, we were able to detect, again, quantify all these amino acids. And the beauty is of this, again, the method or our instrument is that you can see that isoleucine, leucine, nor-isoleucine, alloisoleucine, these are the structural isomers whose identification and detection is a challenging aspects when we deal with the peptides having this structure. So we were able to detect, quantify these amino acids having, which are again the structural similar isomers with good resolution and recovery. Another is the ornithine, where you can see it is present in gramicidin. So yes, we can also do the antibiotic analysis of gramicidin is an antibiotic having ornithine as a within its structure. So yes, such kind of moieties can also be determined, analyzed, and quantified. Then terzeptide, another billion-dollar drug, whose again structural features has AIB as a non-proteinogenic amino acids, along with all the amino acids, naturally occurring amino acids. So yes, it can also be analyzed with our custom standard method. Then beta-alanine, which is a structure-related impurity, and USP 150 general chapter specifically highlights this as a process-related impurity in the manufacturing of the peptide, and its control again can be or its detection can be done with our custom or standard method, Hitachi's method, which we again offer with our previous methods. So yes, AminoScience demonstrates the detection of proteinogenic as well as non-proteinogenic amino acids in your peptides or proteins, even if there is a structural modification like fatty acid side chain or linker if they are present. Now moving forward, till now, or we have seen the peptide sequence up to 40 amino acids. Okay, so what if there are more amino acids, which we call, if the or the definition of the peptide is the amino acids starts from the 2 to 40, then we say it is a peptide. And if it is beyond, then it is a protein. So that's why I have said it's a protein therapeutics. So what we have done is, to again show the robustness of our aminosaya, what I have done is we have taken the latest injection, which is available in the market, 100 IU per ml. So using aminosaya, we have carried out the hydrolysis of this, and you can see here uh, the detection of the amino acids which are present in that sequence. It is a 53 amino acid sequence. So the recovery match, or the expected count match the theoretical count, demonstrating the robustness of aminosaya in the formulation samples also. So this is the ready-made samples which we have shown, which has again the insulin gargalin, talking about the molecule. It has 53 amino acids and with a disulfide linkage. So with the cysteine count, around 6 cysteines are present within the molecule. So we again followed the USP protocol, and you can see the recovery even if there is it is A and B side chain molecules, side chain linkages are there. We were able to also acquire cysteine with good recovery. So aminosaya, I have shown you or demonstrated the capabilities of aminosaya for the peptides and the proteins. Now we'll move towards the quality control applications, like how aminosaya shows the compliance to different batches, or how it can be done, how accurately and efficiently with consistent performance it can be achieved. So let's move. So what we have done is, we were having, so this data is again taken from the consent from the customer. So one of our customer has requested us to have the batch testing for the samples as per the EP protocol, which says that the maximum impurity, individual ninhydrin positive impurity should be 0.2%, and the total should be 0.5%. So we did this batch analysis for batch A and batch B. As you can see in the batch A, apart from ammonia, there is only arginine. So batch A is completely pure. While for batch B, we were able to trace some impurities, and that we identified it was lysine and ornithine. One was process related impurity and another was degraded impurity. So batch B exceeded the limit of EP, which was 1.2%. So it is around 1.5%, and the total impurities were about nearly 3%. So batch A fall in the compliance of EP, while batch B fail. So yes, aminosaya can be used to what we can say, confirm the different, or to test the batches and check the purity of that batches according to the compliance as per placed by the regulatory guidelines. So moving forward, another compliance testing, but that we have done for the amino acid infusion samples, which were used for the mostly for the patients which are to have their level amino acid level higher, or they are having low amino acid content. So what we have done, again, there is a USP compliance for this, and the accepted range of total amino acids present within this sample is around 90 to 110 mg per ml. So there were again different batches with us from our customer, and again this data has been taken from the consent from the customer. So you can see that sample 2 and sample 4, which one of the representative example I have shown, meets the compliance or fall within the USP acceptable range, while the other samples which are present are below the level, or higher. Some of them are below the level, and some of them are were higher. So yes, they such again, the demonstration of such kind of studies can be used mostly for the clinical formulations, where batch compliance is the with the pharmacopoeial requirement is the criteria set. So yes, aminosaya can be applied for this clinical formulation studies, ensuring the compliance of your batches as per the requirement set by regulatory guideline, supporting reliable. reliable quality control in your formulations, or we can say in the drug development. So moving forward to another case study for research and development purpose, as Purusan has said that we provide 7 ports for the buffers, and we provide the recipes also. And with this, I can also add on to it is that with having 7 buffers of different pH range, you can modify the methods also, and use this definitely for the research purposes of your projects. So I have shown one example to demonstrate the aminoScience excellence in the research and the method development also. So what we have done, as you can see here, the standard chromatogram. You can see the 20 amino acids protocol. So what we have done here is we have taken L-cysteine and we have added a green oxidizing agent, and all this analysis or the reaction we have carried in the green medium. So over the period of time, we have observed is conversion from L-cysteine to L-cysteine. Okay, so which is again the dimer of cysteine. So that can be traced over the period of time as we go from 5 hours, where you can see the cysteine peak, to the conversion of cysteine over the period of time with the reaction condition which we have shared. So you can see that we have achieved the conversion of L-cysteine to up to 86%, and the analysis time again reduced to 20 minutes, from 60 minutes to 20 minutes. So such kind of reaction monitoring of the can also be done, as well as the method modification. As you can see, this is a 60 minutes method. We have tried to have it in the 20 minutes. So yes, the method modification and the research purpose studies can also be conducted with the help of aminoScience. So this was one of the examples showing that. Now moving towards the biotherapeutics application, how aminoScience will show its robustness in the biotherapeutic or biomedicine production. So let's move into biomedicine production. So why we have to monitor amino acids, first of all? Because as we know that cells acts as a living factories to produce therapeutic proteins and monoclonal antibodies, which are related to again disease and genes. So to function these cells efficiently, careful culture media is required. That is, the supply of nutrients is the one of the important factor to have a good medicine production, we can say. So the food. So to operate these factories, not only just the temperature, pH, and oxygen is required, but the supply of amino acids is equally critical, as it drives cell growth, metabolism, and overall protein quality. So this makes monitoring of amino acid an essential for ensuring high quality protein and biomedicine production across all stages. So let's understand what with an example what I want to say is, so you can see here the standard chromatographic profile of amino acids, which has again not only non-proteinogenic amino acids but proteinogenic amino acids. So this is our standard chromatogram, which detects 40 or more amino acids along with the proteinogenic amino acids. So the method again, which Purusan has described, the physiological method where free amino acids can be detected. So this is the method where the detection can be reached up to 40 or more amino acids. So till now we have demonstrated 50 amino acids. So when we take the culture medium, on the top chromatogram, we have seen the standard. Now here we have, you can see here, at the initial stage of the structure, there are a lot of amino acids present. But at the top, after 48 hours, over the period of time, glutamine has decreased from high to low. Then alanine concentration has increased. Glycine has again shifted from low to high, and threonine remains stable even after 48 hours. So aminoScience has made this analysis straightforward, giving us a clear picture of the nutrient balance which is present during that metabolic activity in the media. So yes, such kind of analysis or the study can also be done, which will help for the biomedicine production with the help of amino. So moving forward, this concludes our applications, which we have demonstrated with the help of AminoShaya to showcase its capability and robustness using different zone applications, whether it may be peptides, quality control, or protein therapeutics or biomedicine production. So with this, I want to also bring about to a focus that yes, there are these are our valued customers in the pharmaceutical and biotech as well as food and nutrition. So we are not only limited to pharma, we are also the same machines, same methods, and same applications can be demonstrated in the food and nutrition also. And these are our two customers. Another point we would like to also bring about that once our machine is delivered, which provides high precision of amino acid analysis to pharmaceutical and nutrition, we also have a repeat order from the same customers also. So this demonstrate our service, our robustness, our applications, the methodology, and the support which we provide to our customers. So yes, AminoSaya provides a regulatory compliant solutions for peptide compositional identification, batch-to-batch consistency check, impurity and degradation profiling we have seen, and altogether the helps in the sequence verification. So these are some of the examples which we have shown you right now due to time constraint. You can have more applications on AminoSaya at our SI site. If you are interested, you can just go visit and have a login, and you can have more access to applications of AminoSaya. And if you would like to reach out to us, you can reach out to our corporate office, which is in again Mulund, and our technology center, which is in Thane, for your sample related queries, or you want to visit and see the instruments. So thank you for your attention, and we are open for further discussion.
Thank you, Nivedita ma'am and Purushottam sir. We are now opening the session for Q&A. Please use the chat box to share your questions, and our speakers will be happy to address them. We have received one question. Any
Yes.
specific reason for having 7-port immune system?
Yes. As I cannot see the names, thank you for the question. Yes, since, as I said, there is a lot of structural modification happening in the peptide industry as well as protein. It is not only limited to just 20 amino acids. You want to develop your methods, you want to analyze more non-proteinogenic amino acid. In that case, different buffers are required, or different region, as we have seen an image where the ionic strength and the buffer composition helps to elute the amino acids. So these different buffer compositions will help you to detect and analyze the amino acids as per your requirement, whether depending on the matrix. Again, it is not limited to only peptides, it is food and nutrition also, and feed also. So, and another thing also, to our system also take care of the instrument conditioning and the long life of the instrument, we can say. So there are some ports where you can, these are the ports like washing ports, we say. So the inbuilt washing of the column and the instruments also occur with that ports. So yes, this is the purpose we provide you 7 ports.
One more question from this is from Kelkar from Loroli. His
Okay.
question is how sequence is confirmed and how repeat amino acid sequence is confirmed. As on hydrolysis, the amino acids are released from peptide.
Yes. So first of all, amino acid sequence is not, or we can say the amino acid composition is confirmed. Like the sequence count can be verified, as you can see here, one of the example of insulin glargine, which, for example, glycine has count of 5. You will get the count 5. But the sequence verification, like after glycine, isoleucine, that cannot be done by aminoscience. The composition and the quantification can be determined with the help of aminoscience. But the sequence verification is not done with the help of aminoscience. It is one of the orthogonal technique to have the composition verification, detection as well as identification of amino acids. I hope I have answered your question.
Yes, he understood, ma'am.
Yeah, okay.
Any more questions from the participants? Please use the chat box. Uh, One is from Vandana Tiwari. Her question is, can you detect the amino acids also?
Can we detect? I didn't get you.
It's like since it is, you can check in the Q&A box. It
Yes,
is
yes,
written by Vandana Tiwari.
yes. Just yeah. Ma'am, it is can we detect? No, ma'am. Since the technique is there is no chiral column we are using, we can quantify D and L amino acids together, but not separately D and L. But yes, we are working on it, and hope we try to separate D and L. But D and L together can be quantified, but the identification cannot be done.
We have received one more question, ma'am, from Ashish Tomar. We have multiple products in the biostimulant product range for which alginic acid needs to be tested at different concentrations level 0.02%, 0.2%, and 2.0%. Can we do on this? It's in the
Alginic,
chat box.
yes, yes, I'm just checking out. Sir, first of all, alginic acid is not an amino acid. This is a dedicated instrument for amino acid analysis. So, I don't think we can analyze alginic acid. So, if you can give me an idea about the structure, is there any amino group present in it? Because this molecule is specifically, as I said, the amino group of amino acid react with ninhydrine and you get the adduct. Okay, the purple, Rueman purple, or the adduct, we can say. So, alginic acid has no amino group. It is, I think, idobenzoic acid, if I'm not wrong. Uh, so, I don't think it cannot be. It is not detected at all, as it has no amino group. Even if it is having an amino group, like in semaglutide case, we have detected this aminoethoxy AEA, which is a linker, but we were able to do that. So any moiety having primary and secondary amino acids, we can do that, do the amino acid analysis of that, as it is a dedicated instrument for amino acid analysis. I hope, Ashish, I have answered your question.
Yes, ma'am, he noted.
Okay.
We have covered the questions, ma'am. Any other participants would like to share your questions, or we can conclude the session?
Okay. So, Reshma, if there is no more question, then we can certainly conclude this
Yes,
session.
sir.
Yeah,
Yes, sir.
but before that, I would like to extend my sincere thanks to our esteemed speakers, Dr. Purushottam Sutar and Dr. Nivedita Patel, for sharing their valuable expertise and insight on peptides, therapeutic, and amino acid profiling. I'm sorry, I'm not a medico, so my pronunciation of the technical terms may not be as good as you have, but definitely uh, the quality of health system and the health services are equally important as quality of manufacturing. And I'm sure without a healthy or swasth Bharat, we cannot dream of developed Bharat or Atmanirbhar Bharat. Thank you very much. I would also like to thank Hitachi High-Tech India Private Limited, our knowledge partner, for their valuable support in making this program possible. My sincere appreciation to all the participants for your active presence and engagement. Your participation will help us continue building a stronger platform for knowledge sharing and quality excellence across the Western region. With this, thank you once again, and I wish you all a very productive day ahead. Thank you very much.
Thank you so much, sir.
Thank you, Prima, sir.
Thanks.
And thank you.
Thank you
Thank you,
so much,
Nivedita.
sir.
Reshma, you can end the webinar.
Yes, Pushkar.
Okay, thank you, Reshma San, for the coordination and the help. So we
Thank
will
you,
take a
ma'am.
leave. Yeah.
Yes,
Okay.
thank you
Bye.
so much.
Bye. Bye.
Bye.